Thanks so much for that introduction. It's a pleasure to be here to have this opportunity to talk to you guys about my work. I'm a vision scientist, as you just heard, and most of my research has therefore focused on the contents of visual consciousness, visual content in particular. And I think it's fair to say that most of the research in psychophysics, perceptual studies, visual neuroscience, has really probed the observer's conscious visual experience. That's what most people who study vision do. But we have to remember that vision is also used to control movement, often extremely rapid movements. And I think if you look back at the distant past, vision evolved first not as something to represent the world, a perceptual representation, but rather something to control movement. And this is a kind of reductio ad absurdum example of that. Euglena, which, as you know, is a microorganism that lives in ponds. And it's interesting because it has this big flagellum, and when it's in a dark part of the pond, it moves the flagellum a lot. And when it's in a sunlit part of the pond, it doesn't move its flagellum very much, which is a good thing because then it can use sunlight to manufacture food, which it does. And I don't think anybody would argue that Euglena sees the world or perceives the world. You don't have to talk about it that way. You can talk about it entirely in terms of a servo mechanism that, light strikes some photochemical, that changes some activity in microtubules, that affects the flagellum, and that's that. I mean, that's really all you have to say about the visual responsiveness of Euglena. But you can ask the question, what about vision in vertebrates? Does that same kind of argument apply? And indeed it does. If we look back at the initial work by Bekhterev, Vladimir Mikhailovich Bekhterev, who sadly was poisoned by the NKVD at the behest of Joseph Stalin, because he called to some of his colleagues Stalin as a paranoid person. He was consulted as a neurologist to look at Stalin, and he was dead one day later on Christmas Eve in 1927. So be careful what you say, I guess, is the message there. But at any rate, what he did do was to study visually guided behavior in the frog, and he made lesions in a structure in the midbrain called the optic tectum, and he made bilateral lesions, and what he found was that the frog was completely blind, ignoring small prey or large living objects. So that doesn't speak to the issue that I want to talk about, but that was the first foray into vision in the frog. But it was taken up again by David Ingle back in the 70s. I think it's quite appropriate that I'm giving the talk here because David actually spent a long time in Cambridge. When he published this particular paper, he was at McLean Hospital in Belmont, which I had the pleasure of visiting when I spent 2 months here working with him in 1977. I lived over an ice cream shop on Mass Ave. The place was full of cockroaches, but it was cheap. In fact, it was free because the graduate student who lent it to me was in California for the summer. But what David did was to sort of replicate or attempt to replicate Bekhterev. And what he did was to make a unilateral lesion in the optic tectum and to remove the eye on the same side, which meant that all the visual information was going to the damaged tectum. The visual input, as you know, is completely crossed in amphibia like the frog. And so basically all of that visual input was going, not all the visual input, but visual input that was going to the optic tectum found one not there. And he took the frog and he tested it with a fly-like object. And of course, the frog was blind, like Bekhterev said. And he put the frog away. And then 3 months later, he tested the frog. And what he found was that when he presented the stimulus again, now the frog snapped over here. And when he presented it here, it snapped over there. In fact, its entire behavior was mirror imaged. It was actually going to the side opposite the fly, and it never learned to change this. The reversing prism effect, as it were, that was created biologically, the frog was incapable of dealing with it. So why is this? Well, when David looked at the neuroanatomy, what he found was amphibia, of course, can regrow an optic tract when it's cut. And so the optic tract started to regrow, and of course, there was no optic tectum there on that side. So it crossed over to the opposite side, and it laid itself out on a chemomechanical gradient, going to those sites that were appropriate for that side of the optic tectum, which controlled snaps to the other side. So that's why it showed mirror image snapping. Now, does that mean that the frog's perceptual representation of the world is forever mirror imaged? Well, as it turns out, that's not the case. And Engel showed this in a very interesting experiment where he put a barrier in front of the frog, and that barrier could stop here, or it could stop here, or it could go all the way around and stop here. And so when you touch the frog on the rear end, the frog tries to get away, but it doesn't want to jump into the barrier. And notice that the edge of that barrier is in the field in which it showed mirror image snapping. But those lines that you can see represent the actual responses of the frog. It was going the right way, despite the fact that its snapping was mirror imaged. So there can't be a single representation that has somehow been reversed. So what's going on? Well, he went on to show that there's a structure here called the pretectum, which sits at the back of the thalamus, and its projections are still normal. And it, in later experiments, he showed is important for the control of jumps around barriers. And so what you had here was a frog that had, if you like, some independent pathways from the retina to the motor neurons for different classes of motor behavior. So it shows visually guided prey catching through a optic tectum with contralateral projections. Didn't talk about it, but if you loom your hand towards a frog, it will jump away. It's sensitive to looming targets, and that's due to an ipsilateral pathway that leaves the optic tectum. And of course, as we've just seen, you get visual control of locomotion around barriers that depends on the pretectum. So, as I wrote many years ago, vision then initially evolved not as a single system that allowed organisms to see the world, but really an expanding collection of relatively independent visual motor modules. There's got to be some coordination, obviously, but nevertheless, these, in some real sense, are independent visual motor pathways. So what about us? Are we organized the same way? Well, if you look at a sort of simple initial boxology diagram, we seem to be. That is, depends on whether your anatomy, if you're a lumper or a splitter, but there's somewhere between 10 and 14 different sites that receive direct input from the retina, a single synapse input from the retina to these brain targets. So there's the suprachiasmatic nucleus, as you know, which is important for synchronizing circadian rhythms with the local light-dark cycle. There's the accessory optic tract, completely understudied in primates. That's important, we know, from work in birds and rabbits, important for the control of posture. It corresponds to its axes of movement correspond to the semicircular canals. And pretectum involved in locomotion, we assume, we don't know, because it's only been studied in the frog. And of course, this structure here, the superior colliculus, which has been reasonably well studied, that's important for saccades and fixation. And then, of course, where everybody's been, the projections that go, very powerful projection that goes from the retina to the lateral genetic nucleus in the thalamus up to primary visual cortex, and then to the posterior parietal cortex in the dorsal stream, and down into the occipital temporal areas in the ventral stream. So that's been studied a lot, and it's clear that it's important for perceptually representing the world. And these perceptual representations, they really give us a huge advantage, those animals that have this kind of representation, because it allows for a number of things. It allows us to navigate, choose goals, plan ahead, communicate with each other about the state of the world, because we share these representations and we can talk about them. And of course, they can be conscious. And these representations don't have any direct contact with the motor system. There isn't a single set of fibers that leave the ventral stream and that go down to the brainstem or spinal cord or go off to premotor cortex in a sort of fundamental motor control way. In fact, they go to a number of cognitive buffers that then make contact with the motor system for things like speech and planning and so on that might engage the motor system. But at the same time, there's increasing reliance on skilled actions, actions that we engage in addition to the ones that are sort of shared with many other animals. But we play hockey and we make tools and we drink from wine glasses and so on, where there is a lot of control that comes from the cerebral cortex to learn and engage in these very skilled actions, many of which are very rapid. So all of this led, as some of you know, to an idea that David Milner and I had back in the early 90s for two visual systems in the cerebral cortex of the primate brain. One system that allows you out there to recognize the objects, the cup and the pen and or pencil rather, and the wristwatch and the hand and so on, and to know it's a picture. It's not real. You know that by looking at it. But the actor in the picture is doing something very different. And what I would argue, and indeed what David and I argued, is that the visual system that allows you out there to see those objects and to recognize that it's a picture is fundamentally different in both functional and neural terms than the one that allows the actor in the picture to reach out and pick up the cup. That fundamentally different neural pathways are engaged and different transformations are performed. And we, over the years, carried out a number of experiments that strengthened this point of view and suggested that you could map this functional distinction onto the two neural pathways that leave early visual cortex and go up to the posterior parietal cortex, the dorsal stream, well poised for the control of action, massive projections down to the brainstem, to the pontine nuclei, lots of reciprocal connections with premotor cortex. So it's great shape for actually controlling action. And the pathway that goes ventrally down to the infratemporal cortex that's important for perception, making contact with mesial temporal structures involved in memory, social interactions, and so on, projections up to ventral prefrontal cortex, important for decision making, and so on. So again, their connectivity patterns and so on set them up for transforming visual information in different ways. Now, I don't want to say that that's somehow all that the posterior parietal cortex does. As this wonderful review by Dwight Kravitz and co. show, you missed your picture, that there are other things that are going on as well. I mean, in addition to visually guided action, there's spatial memory, navigation, and so on that the structures in the dorsal stream are involved in. But I want to suggest that even here, a lot of those cognitive functions, like working memory and attention, likely evolve from visual motor processing. And in another talk, I can develop that argument, but if you want me to talk about it, I can talk about it later. So the Rosetta Stone for us then, for understanding this was a patient DF, who had carbon monoxide poisoning which left her with these bilateral lesions in her ventral stream in an area called LO that Nancy Camisher and others have shown is very important for the recognition of objects. And indeed, she had some real fundamental problems with form. She was a visual form agnostic. And she, this is a classic matching task where you ask someone to rotate their hand or a card in their hand to emulate the orientation of the slot in front of you. And she's unable to do that. She can't verbally tell you, and she can't match either. She has no idea. But you ask her to post the handheld card into the slot, and she's really good. So she's able to post the card into a slot that she can't perceive. So somehow her hand, or at least the structures controlling her hand, know something that she doesn't. And if you look at patients who have damage in the dorsal stream, DF had ventral stream damage, I'll remind you, then what you find is that the patient here can describe this is lovely work by Marie-Therese Pernin and Alain Viguero, showing that even though they can describe the orientation, it's slanted or it's vertical, horizontal, whatever, they can't post their hand into the slot unless they make contact with it. And then, of course, they can do it using somatosensation, but they can't use vision to guide their hand. So, there's a lot of evidence from neurological patients that point to this distinction. There's, since the development of the Evart's technique for recording from awake monkeys, there's lots of work by Mountcastle and his descendants looking at visual motor control in the dorsal stream in primates, non-human primates. And there's sort of a controversial cottage industry looking at visual motor psychophysics, looking at the fact that in many experiments that we've carried out, people who reach out to pick objects in the context of an illusion, their hand is not fooled by the illusion. That is, the context doesn't seem to affect them, even though they clearly see one object as being bigger than the other. Their hand doesn't reflect that behavior. And of course, with the advent of fMRI, which came much later than when we studied DF, there's been a considerable amount of confirmation of the idea that the dorsal stream is involved in visual motor control and the ventral stream in perceiving all kinds of things. And this work on DF that you see here was led by Jodie Cullum, who did her PhD with Patrick Cavanaugh at Harvard and is now a full professor at Western. We managed to keep her despite all kinds of offers that she got from other places, so I'm grateful for that. So that's all ancient history. So I hope that I've convinced you a little bit that this idea of two visual systems, one for perception and one for action, is at least viable as a kind of first cut in trying to understand the functional distinctions between different visual pathways in the brain. So our contribution then, the contribution that David and I made, was to emphasize the role of vision in the control of action. And, you got to remember that the natural selection can only really work on the products of action. It doesn't matter how well you think about things. It's what you do that really matters. As one of my old supervisors, Gordon Mogensen, said, you can do all the fancy skating in the world, but what counts is a puck in the net. So you have got to act on the world in order to give something for natural selection to operate on. So thinking and perceiving and remembering are all handmaidens, really, to action. Brain is there to control your actions, and that's why it evolved. So the primary division of labor then between the dorsal and ventral stream is that the ventral stream identifies goals and together with prefrontal cortical areas and so on, plans and appropriate action. And the dorsal stream, in conjunction with a lot of circuitry in premotor cortex, basal ganglia, midbrain, brainstem, programs and controls those actions. So when you're trying to understand what's going on here, the two visual systems hypothesis then conceives of the role of the two streams as defined in terms of the consumers. You're going to transform that information from the eye into something that the motor system can use, or to construct a percept as a kind of perceptual foundation for cognitive functions. And it kind of echoes what we said many years ago, David and I, when we wrote the first iteration of the two visual systems in Trends in neuroscience, that the differences between them are largely a reflection of the specific transformations of input required for perceiving the world versus acting on the world. So just to be, completely transparent about what I'm trying to say, the ventral stream converts signals from the eyes into visual percepts, and the dorsal stream converts signals from the eyes into goal-directed motor outputs. So the visual information then used by the dorsal stream, or the information from the eyes, is, according to the model, not perceptual in nature. It can't be accessed consciously, even in principle. Like, it's not as though it's somehow buried there, and if only you could get at it, there would be a percept there that's controlling your movement. No, it's being transformed directly along with lots of other things, somatosensation signals and so on, into a motor act. But it somehow seems evident, the actions that we perform on these visible objects makes use of the same representation that we use to see it, that somehow we're using our vision of the cup to control our movements towards the cup. And, Andy Clark, amongst others, has said that this is a kind of grand illusion. That's you've got an assumption here, this assumption of experience-based control. Yes, you identify that as your coffee cup and so on, but the fundamental act of picking it up is something that's happening independent of that experience. So, this is world's simplest diagram, but, on one side, you've got things that are happening in the brain that are inaccessible to consciousness. And then on the right side, things that are accessible to consciousness. And you've got a lot of inputs into the dorsal and ventral stream. Many of them are shared in the sense that they are the same kinds of information. But when the percept somehow evolves in the ventral perceptual stream, it can be conscious or it could be unconscious. I mean, it could be masked or you could be not attending to it or whatever. But in principle, it can be conscious. And as I've said, the visual information going into the dorsal stream can never be accessed through consciousness, but you're conscious of the act you perform. I mean, you have a sense of agency that you're performing that act, but it's not visual. But it's what the eye information is doing to control that behavior, but it's not perceptual image. So it's important to remember that ultimately, though, both streams contribute to the control of action. They have to, because otherwise they wouldn't have evolved. I mean, they have to make some difference in the world for natural selection to operate. And we can perhaps see the contributions of the two streams to the act of reaching out and grasping an object. It looks like she's taking a selfie, but she's actually picking some grapes there. And if you look at when she reaches out to pick up grapes or to grasp a glass of wine, the dorsal stream will provide some fundamental information about the size, the overall form, the location, the orientation. That information the dorsal stream can do quite well, as we know from the work of DF. She can use that information to control her grasp. But the ventral stream also contributes information about the weight, about the compliance of the object, about its fragility, about how slippery it is, and its temperature, whether it's hot or cold, and its function. Most importantly, its function. So, the ventral stream then enables us to really act on the basis of information from the past. The dorsal stream is working in the here and now. It's a sort of just-in-time computation. But the ventral stream is bringing to bear all kinds of information from the past about through associative learning. You learn about that eggs are fragile, that feathers float, and that gold is heavy. You learn those things, but you use that when you pick them up. But that's referring back to things you've learned on the basis of ventral stream information, and also about the function of objects as well, like hammers and bananas, teacups, and so on. You know what to do with them. So that helps you select the part of the object that you're going to grasp, even though the grasp itself directed to it is controlled by dorsal stream information. And you can see this in things like end state comfort that David Rosenbaum, a term that David Rosenbaum developed, where when you're reaching out to pick up a screwdriver to use it, you show this awkward grasp if the business end is facing you. You turn your hand like this, so you can use it to screw the screw into the wood. And it's an awkward grasp, but it's ready to use. You've got end state comfort. Now, there's a very efficient grasp, but it's really leaves you poorly prepared to use the screwdriver, unless you're planning on murdering somebody. It's a different kind of action that you might perform with that object. And what's interesting is that DF, she grasps screwdrivers exquisitely, but wrongly. That is, she reaches out to pick them up as though they're a branch or something. There's no notion until she finds out. And of course, then she's better equipped to deal with it. But if she doesn't know, then she picks it up in the way that the person at the bottom of that is picking it up. Okay, so all of this seemed to resonate to some degree with a recent paper, some of you know, that came out just this year, I guess. Did it come out this year, Matthias? Yeah. Which my colleague, a colleague of mine, Brian Cornell and I were pleased to be able to write a commentary on this notion that consciousness is slow and it takes time in order for conscious perception to emerge. And as they say in their abstract, it can't be used, therefore, for the online control of action, at least rapid actions like the cheetah chasing the antelope that I indicated at the beginning. And that they argued that this consciousness emerged, therefore, as long-range visual horizons emerge as a possibility. So in the ocean, it's pretty murky, and you have to react quickly to a predator if you're a prey animal. Whereas you've got time to plan. If you see a wolf way over there on the other side of the woods, you can maybe go on the other side to avoid interacting with them. So you've got time to use consciousness, and it can evolve in order to avoid being eaten. But of course, the two visual system story that I've been telling and that Matthias and Steve used was really, it was all on there. It was pretty corticocentric myopia, you might say. With the advent of fMRI, we've learned a ton about the cerebral cortex, but not too much about stuff underneath the hood, as it were. Because, of course, it's difficult to do so. Recently, people are doing it. That's for sure. In the last decade, it's been quite a bit. But initially, it wasn't something that people. It was like looking. I think. Excuse the analogy, Nancy. Looking for the key under the lamppost. I mean, you're missing a lot because there's a bunch of stuff going on in the subcortex that has been essentially ignored. Now, I'm guilty as anybody. I'm a neuropsychologist by training, and the people I study are people who had damage to their cerebral cortex because they're still alive. There aren't very many cases of people who have strokes. It happens, but strokes in the brainstem or lower down the subcortex, they can be lethal, and people are decimated or dead. And so, therefore, the neuropsychology industry, as it were, is built on people with lesions of the cerebral cortex. But as we've already seen, there's all this stuff projecting from the eye to other structures in the brain. And one structure in particular that has been studied to some degree in animals is the superior colliculus, which we know is important as the optic tectum, as it's called in amphibia. In behavior, as we've already seen, in the control of visually guided movement, like snapping at. I'm getting confused now because I realize I have two slides here out of order, but it's known as the optic tectum in amphibia. In humans, it's the superior colliculus. It sounds like a rank in the Roman army, but it's that structure on the top of the midbrain, part of the quadrigemini, and visually guided behavior in the frog, mediated by the optic tectum. So just so you see it work here in slow-mo. Here he goes. By the way, there's no anticipation of where the fly is going to go. It all happens so fast that the frog just goes out there with its big sticky tongue and gets the fly. It doesn't go ahead of where the fly is flying or anything of that kind. It's just so fast. And similarly, when you present a looming stimulus, which I talked about before, the frog jumps away efficiently away from the looming target. Now, in humans, in primates, the superior colliculus gets direct input from the retina. It also gets indirect input from the visual cortex, and it clearly, as we all know from textbooks, it plays a critical role in the control of saccadic eye movements. But it's important to remember it sends information not just to the motor neurons controlling the eye, to the oculomotor muscles, but also to the neck muscles, and in quadrupeds, and indeed in us, to the forelimbs, and maybe to other limbs as well. It's not well studied, but there's clearly input because orienting involves moving, not just your eyes, but your whole body, your torso, and so on. And of course, it also sends input up to the cerebral cortex via the pulvinar and the lateral nucleus. So work from our group suggests that it plays a role in the initiation of rapid reaching movements towards visual stimuli. It's as though it's it's important for orientation, and the limbs are involved in that, but that's somehow been co-opted in the control of reaching. When I say our group, I don't mean our group, I mean Brian Corneal, who is an expert on this. He's been working on primates and laterally on humans. And it's been a great collaboration because he's a real expert on motor control. I'm a vision guy. I dabble in visual motor control, but I'm not deep, into the ins and outs of how motor programs work. But Brian is. And to do this, we use a robot arm, Kinarm, Canadian company. And the way it works is you have a manipulandum that you hold, and you look into a semi-reflective mirror, and you can see targets that appear down here, and you can chase them and do things with them. And the great thing is you can measure the forces that people apply, not just the kinematics. You can measure the forces that they apply, which happen early. You can also wire them up so you can record EMG. You can record from the muscles. You can use penetrating electrodes, which we do with some brave graduate students, and you can use surface EMG. And measuring activation in this way, EMG and forces, gives you way better information about the timing of things, because you know exactly when the signal gets to the muscle. You can see it in the EMG. And of course, you can see it immediately in the force that the muscle contraction produces. You don't have to rely on videotaping or video recording the movement or using iReds or other methods. So here's a standard example. This is a pro-reach and anti-reach task. It's kind of like a pro-saccade, anti-saccade that people have studied a lot in monkeys, where you get people not to reach towards the target, but away from it. And of course, that's hard to do. You have to learn to do that because your natural instinct is to point to the target. So you've got to inhibit that, and you've got to remember the reprogramming that you have to do to mirror that. It could be a 90-degree one, and people do that as well, but we just did a simple anti-reach. It wasn't done like that. It was done by holding the manipulandum underneath the mirror in which you see the targets. So this is a complicated slide, but it's important that I go through it so you understand what we're doing here. So we're recording from the pecs here, and here. And when you record from the pec here, it contracts when you make a movement like this. It's got the manipulandum, make a movement, it contracts here. And what we've got here it's a raster plot, not of neuronal signals, but EMG, muscle fiber firing. And it's stacked from the slowest movement to the fastest, and those are little white squares that you see. So slows up here and shorts down here, short latencies down the bottom. But what you see here outlined in the red rectangle are what we're calling the express visual motor response, or the EVR. Notice that it's linked to the target, not to the timing of the movement itself. It's always occurring at 80 milliseconds after the target's presented in this field over here, because that's the PECT recording from. It's in this field where you normally make a movement towards the target like this. What you'll notice, I'll just anticipate what I'm going to say later, that it gets stronger, its amplitude gets bigger, the faster the movement. So its latency is not related to the speed of the actual movement that you can see here, but its vigor is, its amplitude is. So the larger the response of the EVR, the faster you move your hand. So what happens if you do an anti-reach? Now you're supposed to be moving over here when the target occurs here. It still fires to that target in the pack. It still fires to it, but not as much. It's been dampened somehow, but it's still firing. And what you'll notice is that, as I've already said, the magnitude of the EVR is correlated with the latency of the response that follows, but the correlation is opposite for pro and anti-reaches. That is, the stronger the amplitude is, the faster you reach in a prosecod, and the stronger the amplitude is, the slower you move in the anti-reach. It's as though if you've got a big signal, somehow it takes time to overcome it in order to reach away from the target. And in fact, you can see that when you get really massive activation of the EVR, you make errors. You go towards the target instead of away from it. So a lot is revealed here about what's going on with the spear colliculus in the control of a reaching movement that we didn't really understand until we looked directly at it in this kind of task. And this graph just sort of outlines everything I just said. I won't spend time on it, but we can then sort of raise an interesting question. Where does the EVR originate? We think it's the colliculus. I have to say, we do not have direct evidence yet, but we have a heck of a lot of circumstantial evidence. And we're gearing up, or Brian's gearing up, to do this in the monkey arm. He has done it in the monkey neck, so that you can find EVR in the neck, and you can record or microstimulate, and you can get the appropriate relationship between what you see in the colliculus and what happens with the neck, just like you can with the eye. The arm he has not yet done. So where does it originate? Well, there is a lot of work going back to the 90s, actually, by Klaus-Peter Hoffmann and others that show that if you record from neurons in the colliculus, you can see reach-related activity in that colliculus that's correlated with contractions of the muscle. So you can see that these left neurons, left collicular neurons here are firing, I'll leave it there, are firing, and their profile matches what you see in the muscles, and they anticipate the muscle activity. They're ahead of the onset of the movement. And these reach neurons are found in the intermediate and deeper layers of the colliculus. There's also more recent work by Peter Hoffmann showing that if you microstimulate these neurons, you record from them and then microstimulate them, you can drive the arm movements in both trained and untrained monkeys. What's interesting is that when you do with trained monkeys, they move towards the target they've been trained on when it's presented, and not so much, obviously, in the untrained monkey, but you get movements in both cases. You can also use chemogenetic silencing of arm movement neurons in the mouse colliculus. So the mouse has become a real model now for the colliculus, amongst other structures in the visual system. And this is a task in which the monkey shows how primate biased I am. When the mouse reaches through the slot to get a drop of water, it has to use one arm to do this, and it brings the drop of water back to its mouth. And you inject a viral vector that contains something that can turn off the cells that you've recorded that are related to reaching. And then you inject intraperitoneally a drug that activates the inhibition, and you can shut them down. And when you do this, you inject the vehicle. These are tracking of the hand movement here. And you can see when you just put the vehicle in, nothing happens. When you put in in the actual drug, what you can see is that you get this incorrect trajectory. They go to the wrong place. And then you do it the next day with just a vehicle that contains the drug, and again, the mouse behaves normally. So there's that. And this just shows a graph of what I just said, that you get inaccurate aims when you silence those neurons in the colliculus. There's actually fMRI now, this is actually quite ancient, 2012, showing that unlike saccadic neurons, the neurons that are related to reaching are lateralized, so that the left colliculus controls the right arm of the peer and vice versa. So there's lateralized control of arm movements through the colliculus. So going back to the question, where does the EVR originate? Well, I think there's good evidence that it's collicular. It's really short, 80 milliseconds in the human. There's no way that the cerebral cortex is going to actually produce an EMG signal in the muscle from signals arising in the cerebral cortex. The activity in neurons in the monkey and the mouse are correlated with reaching movements. If you stimulate the colliculus, you can activate reaching movements in the monkey. If you silence the neurons in the mouse, you can displace the reaching movement. And in fMRI, as I've just shown you, there's lateralized activity when people make reaching movements in the magnet. So all of this, I think, and other evidence I haven't mentioned, suggests that the EVR originates in the colliculus and likely reaches the arm muscles via the tectoreticular spinal tract. But we all know that damage to the posterior parietal cortex produces optic ataxia, which is a problem in reaching towards targets. And this term, optic ataxia, was actually coined by Balint. Germanification of his name is Rudolf Balint, who published in German in 1909. And I think it's fair to say that the neurological community until recently really didn't know what Balint had to say because it wasn't translated until 1995. And the Balint syndrome we do know, which is this inability to perceive the visual field as a whole, something like nausea, difficulty in shifting eye movements or attention, oculomotor apraxia, and inability to move the hand to a specific object using vision. That was oculomotor apraxia before that, optic ataxia when you're talking about the hand. Okay. But as I said, it wasn't until Monica Harvey, who was a PhD student of David Milner's, translated, she's a German by birth, and she translated Balint into German in 1995. And I think that it's fair to say that the Anglo-American neurological community really didn't know what he had to say until that translation was published. And what did he say? Well, what he didn't say was what Gordon Holmes said. And Gordon Holmes had a much greater influence on the neuropsychology and neuroscience and neurological communities. He talked about it as a deficit in spatial processing. It was an overarching deficit that affected both your behavior with respect to knowing where things were out in the world and also controlling your movements in the world. He spoke eloquently, he wrote brilliantly, and you can see echoes, I think, of his influence in things like the Unger, Leiter, and Mishkin story on the dorsal and ventral stream. But if you read Balint, you notice something very interesting. When asked to grasp a presented object with his right hand, it was a one-patient study, a case study, he would miss it regularly and would only find it when his hand knocked against it. The motor rather than the visual disorder was the dominant one, since all the movements performed efficiently with the right hand were executed perfectly or with very little error with the left hand. Only movements that required visual control were faulty. So even though the right hand couldn't find things, if you touched his arm, he could touch it with his right hand. It wasn't a motor problem. It was a visual motor problem. So there's no way that some kind of general deficit in spatial processing can explain this. We had neglect as well. And if you look at the lesions that you can see that there was an asymmetry in the lesions on the left and right, one affecting the right hand for optic ataxia, but not the left, and the other producing neglect. So here's the patient.
Can you see it?
I can see it, but I can't tell. I can't reach it.
You seem to do better with your left hand. And take a crack at it up here, if I hold it up closer. All right, how about now that you touched it with that hand, can you reach with the other hand and grab it? With your right hand?
With my right hand?
Uh-huh.
No, I can't.
So that's a kind of typical thing that neurologists see in such patients. This isn't a balanced patient, by the way. Video wasn't invented then in 1909. But Bob Rafal at Bangor lent it to me. And it's a nice demo. It's a very similar patient. And there seems to be a right-hand thing, in humans at least. That what seems to happen is that if you have damage to the left hemisphere, it affects the right hand in contralateral space from the damage, but the right hand also has problems over here. Not true of the left hand. It's all over there, but not here. So really interesting. Okay. So there's all these neurological studies that show that the posterior parietal cortex plays a role. And of course, there's a ton of fMRI studies as well. I've just got a few here from our own lab that show that along the interparietal sulcus, there are structures that are involved in that are activated when people reach for objects in the magnet. Again, work by Jody Cullum. So, how am I doing for time? Oh, I'm almost done. So, no talk would be complete without a cat video. So here we are. You've all done this if you have a cat, chasing a laser pointer. Right. So, this is a jumping target task. And the jumping target has been studied a lot. And it was first brought to prominence by Marc Jean-Roux, who I had the pleasure of working with many years ago. And here's the task. You're reaching for a target, it suddenly jumps unpredictably, and it's effortless. You just correct. It's really easy. And you ask people to do one of two things. On some trials, they corrected, the way you just seen. On other trials, they just produced an explosive vocalization when they saw it jump. Jump, you say, or something in French. And the motor correction latency is about 120 milliseconds, really fast. You can see it in the trajectory, but it takes you 420 milliseconds to actually respond consciously, if you like, that I saw it jump. And indeed, back in '83, just after I got married, we took a whole year off and traveled around the world, but we ended up in France, and I worked with Marc, and we did an interesting study led by Claude Prablon and Denis Pellisson, where we were interested in the visual information or not that's used to control the hand when you reach into a target. And we demonstrated that you didn't need it. You could use proprioceptive information or other kinds of things to control your movement in the absence of any vision of your hand by jumping the target during a rapid eye movement. So they looked and reached, but secretly, when they were moving their eye really fast, you jump the target to a new location further away or slightly in. And that was interesting that they did those corrections. But was even more interesting, we didn't expect it, but then we studied it, was that nobody saw the target jump. And in fact, they found it hard to believe, these are people in the lab, that the target had jumped. And so we tested them on a two alternative force choice, and we found that they could never detect it. So the neural mechanisms mediating the perception of target position can be dissociated from those mediating visually guided reaching movements directed at that target. In fact, this was expanded by Yves Rossetti later, who also worked with Marc, saying that that was the very particular role of the posterior parietal cortex. It was really important for the control, online control, the automatic pilot, controlling your movements. And so we thought that would be a really interesting thing to study in the context of the colliculus. So we had a jumping target task that was actually done by David McHale, now off in medical school, sadly. Oh, I guess everyone has to leave the lab, but he's gone off to the University of Toronto. And we had a jumping target task where there was no jump trials in which they reached directly towards the target. And then there, on 30% or 33% of the trials, the target jumped unpredictably to a new location. But we had different task instructions. They either just follow the task, really easy to do, or they had to stop, hard to do, hard to stop. It's what we called in the lab the oh shit phenomenon, because you make the movement and you say, darn it, I wanted to stop, but it was hard. Or you simply have them ignore it. That's easier. It's not perfect, but it's easier to ignore it. And the reason it's hard to stop is that that's a signal to stop. Whereas in the ignore condition, you can just ignore it. You don't have to attend to it, whereas you have to attend to it in the stop condition. And what we found was, here's the EVR in this period here, and these are average traces of an EMG in the muscle. You can see it's bigger on the follow tasks and smaller in the stop tasks because it's being dampened somehow, presumably by cerebral cortical mechanisms that are task-driven. And it's quite suppressed in the ignore task, which is, of course, easy to do because you don't have to attend to it in order to ignore it. At best, you don't. And this just shows what I've just said, that what you've got is faster, bigger movements in the EVR epoch when you follow, smaller ones when you stop, and even smaller ones when you ignore. And so the automatic pilot then seems to be driven by that EVR. But one of the reasons people liked the automatic pilot was that it was so fast, had this short latency. But of course, that's all kinematics. All you're doing is recording the actual things you see on the video or on the recording from the iReds. You're not actually measuring the forces or the EMG. And here's some forces. If you look at movements from rest versus online correction with the kinematic measures, the video measures, what you can see is that you're slower to get moving than to make an online correction. By considerable margin, 50 milliseconds or so. But if you actually measure the forces in the manipulandum, they're the same. They're early, 141 when the movement from rest. But of course, you've got a biomechanical problem. Your hand's still. And now you've got to get it moving. But if you're moving and you jump it, you've already overcome a lot in order to keep moving. And so all this kinematic stuff that I and others have been doing for years is missing a lot because it's not measuring the actual forces and EMGs that are the temporal parameters that are there in the muscles from these control systems in the brain. So, and this is my penultimate slide. This is a nested control system, if you like. We've got visual control of arm and hand movements made up of subcortical and cortical pathways, but they contribute to distinct phases of the muscle recruitment. We got this very fast phase that's generated by the colliculus and by the tectoreticular spinal pathway. It's got the huge advantage of being fast, but it's dumb, right? It's inexorably stimulus driven. You've got to suppress it sometimes. And that's where cerebral cortex helps because you can use conscious and deliberate task setting via these cortical networks to dampen or liberate the EVR. So it's true that these nested control systems benefit from the conscious visual perception that Matthias and Steve mentioned in their brilliant BVS target article. And they evolved, no doubt, in these long-range sensory horizon conditions. But we're still using, however, these ancient systems. It's as though we're co-opting them through these much more recently evolved systems to control our behavior. So in short, we humans enjoy the best of both worlds, the ancient world and the more recently evolved one, which was the title of my talk. So we're now planning to look at patients with damage to B1, patients with damage to motor cortex, to see if we can still record the EVR, even though the people may not be able to move or even see the target. And that's it. Thanks very much for your attention. Sorry, I took a bit of time.